The state-of-the-art aerobiology TB research facility is pinning down how tuberculosis is transmitted. According to UCT Professor Robert Wood, TB infections in the community increase at an astronomical rate.
We believe that the time of going to primary school at the age of 5, 20% of these populations are infected with TB. And just before puberty at 14, 50% of them are infected with TB already. And by the time they leave school, 70 to 80% of them are infected with TB. And we think fundamentally we have to control that.
Wood says they're focusing on drug-sensitive TB patients. This will help identify what he calls super spreaders, a key to curbing the spread of the disease.
This is the first stage in our sampling. We put a TB case into there for about an hour, and we monitor all the environment around them, and we capture the air that they breathe out over that hour into a small quantity of liquid.
Once this liquid is captured, the sample is separated into smaller parts in which TB particles can be identified.
Here we take the solid component of that, which is spun down, and we put it onto what we call a matrix, which is a slide with thousands of very small wells, and the organisms get captured in one of those wells. Then we look at the fluorescence of that organism and can study and focus on a single organism. This is really the sharp end of science nowadays, is to be able to do single cell analysis, to be able to look at the characteristics, what we call the phenotype of the organism, and to see how it's adapted for airborne transmission.
Once the process is refined, Wood hopes to test at least 2 patients a day. The research is set to continue for at least another 2 years. Stefan Goosen, Cape Town.